So welcome everyone to our continuing webinar series for the GDC. I will be your host today. My name is Brandon Chan. I'm a technical business analyst for the GDC data portal. Last month, we covered the data transfer tool for GDC, and this month we're actually going to go back to the web portal and show you a very basic use case on how you can build and analyze a cohort and download some data from that cohort. So just a very quick overview. If you have any audio visual difficulties, please do reach out to the moderators. They will assist and stop me anywhere as needed. What we'll be covering today is, first I'll give you a quick overview of what we intend to do. We'll go through a very simple use case, and then we'll get into the nuts and bolts of it. So building your cohort, analyzing it, and downloading any associated data. And then finally, we'll have a small open period at the end for any questions that you might have. So what do we plan to do today? Well, I know there is a wide range of users in our audience today, ranging from maybe some basic first-time users to some more advanced power users. But essentially, at the end of the day, the GDC data portal provides very easy-to-use, flexible, and powerful tools for you to build a custom cohort of cancer cases. Analyze those cases with some out-of-the-box tools that we have. And then if you want, download some of that relevant data, some of that raw data, so that you can continue further analysis on your own, offline, maybe with some other third-party tools. So our goal today is really to show you how to perform this process with a very basic sample use case. So here you see a small, nice little picture of what that process looks like. First, you will be wanting to explore the data that's available in the portal. You'll want to see what's there before you build your cohort. Once you filter down and reduced to the set of data that you want, then you'll build that cohort and you'll save it. Once you've saved it, then you'll want to analyze it with the tools that we have available. And then finally, as part of that analysis, maybe you want to dive deeper into that data, and then we will show you how to download that. Now, before we start, I do want to go through a few definitions just so that we are on the same page. It's always good to use the same language. I may be using some of these words throughout the webinar. So just want to make sure we are of the same understanding. So the first word that may come up is program. So program is really a cancer genomics program that has supplied data to the GDC for exposure in our data portal. So for example, a very well-known one, of course, is TCGA, the Cancer Genome Atlas. Next is the concept of a project. Project is it's a specific project within a program, and it may focus on a specific area of study or research goal. Again, using our TCGA example, we might be looking at TCGA BRCA for breast invasive carcinoma. And then some concepts within these projects. So a case. A case is basically the key concept in our portal. It's a collection of all the data related to a specific patient in the context of a specific project. Then we have genes, so specific cancer genes of interest that affect one or more cases in the GDC. A mutation, so a specific cancer mutation on a gene of interest, again affecting one or more cases in the GDC. And then two things that will come up quite often today. First is cohort, so that's the whole reason we are here today, to build and analyze a cohort. For the purposes of this presentation, a cohort is a specific set of cancer cases that a user wants to group together for further data analysis and exploration. and then a set. So what is a set? A set is basically a specific group of things that you can save in the portal for future use. And those things that you can save are a group of cases, a group of genes, or a group of mutations. So for example, if we relate this back to the concept of cohort, I don't want people to get confused with these two terms, but essentially a custom cohort is created by saving a set of cases in the portal, or a case set. But for today's purposes, we'll mostly just be sticking to cohort because that is what we're trying to accomplish today. But you'll see how these things interplay as we go through. Now, I know everyone's eager to actually get into the UI and play around with the interface, but first I want to go through the use case we're going to walk through today. So we'll be performing a very simple use case just to illustrate how easy and flexible the portal can be, even for lay users. Again, I know there's a wide range of expertise in the audience. But I'm hoping that this simple use case can not only, educate and inform, more basic or first-time users, but then also give advanced users, more ideas of how they can extend and tailor such an example to more of their specific needs and requirements. So the specific case I've selected, I've actually decided to use myself as a guinea pig. Because I am a business analyst by trade. I do not have an extensive, scientific or bioinformatics background, as I'm sure some of you do. But I think it helps illustrate that even a layperson can get a hang of this portal quite quickly. So specifically for myself, I decided to use a fairly personal example. Like many people in this world, I myself have been affected by cancer in my personal life. So my mother did have lung cancer. Specifically, it was an adenocarcinoma, and she was diagnosed when she was age 45. So, perhaps I am a person who's a little bit interested in, that demographic of females 45 and up with lung cancer, adenocarcinoma. What are some of the trends? What are some of the clinical properties that come up in cohorts of this nature? So what we're going to do is we're going to build that cohort specifically in the portal. Then we'll do a very quick comparison of that cohort with a similar cohort except for males. We'll do a quick survey of some of the major clinical properties for that cohort. And then finally, we will download some raw clinical and genomic files and show you how that is done. Okay. So great, now we can go into the UI. I'm actually going to stop the presentation so that I can actually go to the browser. Okay, so let me just minimize this. Just do a quick check here. Everything okay? Everyone can still hear and see the screen all right, I assume? Okay. So I am going to go to the portal. Okay. And what you see here is the home page, the home landing page for the GDC data portal. Now, the key area of the portal is the exploration area. So you'll see a lot of different navigation links up top. I won't be covering all of those today, simply because we have webinars in the past and some in the future that cover some of these areas. So I do encourage everyone to check out some of our previous content. But today we'll be focusing mostly on exploration and analysis. So let's say I come in and I say, I want to build this cohort with these certain conditions. So I'll enter my exploration. So before we start, let me explain to you the structure of the exploration page. Again, I know some people may already be familiar, but I think a refresher is still worthwhile. So what you see here on the left-hand side is what we call filters. So the filters allow you to basically choose certain properties that you want to examine. and narrow down the data that you see to exactly what you want. Now there's 4 types of filters to help you narrow down that information. So first is the Cases tab. This is sort of very high-level information about the type of patient that you want to find. So you'll see important concepts like the primary site of the cancer, the genomic program that you may want to look at, the specific disease type, so on and so forth. The second tab are specific clinical properties that you want to look at. So earlier, we mentioned, gender. So I could use this to pick, female or male, and you can see there's a wealth of clinical properties that we can go through, and we have a lot of documentation on what those properties are. But there are some major categories: basic demographic information, diagnosis information, treatment information, and exposure information that patient has gone through during their lifetime. Again, I'll show you a more specific example. The third tab is the Genes tab. So this is useful, for example, if I wanted to, maybe I'm interested in a very specific set of genes for my cohort. Bronchus and lung cancers, TP53 is a gene that has some significance, so I may want to narrow down my results to that specific gene. Similarly, for the Mutations tab, I can narrow down to a specific mutation or maybe a certain type of mutation. So for example, maybe I am interested in TP3 mutations where there's a missense consequence. So I can use those filters to narrow it down. So again, just to summarize, these filters are to help you narrow down your search results, essentially. What you see in the middle area, see this table right here, is basically the results of your filtering. Again, you'll see multiple tabs here, and I'll go through them one by one. The first tab is the Cases tab. So this is, as you narrow down your search, which patients across the GDC meet the criteria that you have filtered against. And you can see there's some relevant columns. I won't go through all of them, but it gives you some high-level information. What is the ID of that patient? What is the primary site of their cancer, their gender, and very importantly, the number of mutations and number of affected genes for that patient. And you can see that the results are actually ranked in order of the most number of mutations. So you can see this first case here, 1,856 mutations, because generally we would be more interested in patients with more mutations. The second tab is a Genes tab. So what are the genes that meet the search criteria? Similarly, the Mutations tab lists mutations in the portal that meet the criteria. We won't be focusing too much on these two tabs today because we aren't building a set of genes or a set of mutations. Today we are building a cohort or a set of cases. The last tab we have here is OncoGrid. I won't get to this quite yet. When we get into the analysis, then I'll cover that a little bit more. But this is the basic structure of the exploration page. It is really, at the end of the day, to help you explore the GDC data and narrow down to what you want. So now that we've covered those basics, we can actually get to building the sample cohort that I was talking about. Now, one thing that you might notice up top, you can see in this little blue area, there's actually a line of text that says, "Is cancer gene census true?" So this blue area up top, it actually shows you the query that you're building as you're adding filters. So you're probably wondering, why is there already a filter by default? Well, this is actually a convenience that we provide users. So one feedback we've had is, people generally are quite interested in, by default, the genes that are part of the cosmic cancer gene census because these genes have been proven to have high impact on many types of cancers. So by default, we apply this filter just to narrow that down to you can see there's 576 cosmic genes. So we kind of narrow that down by default just to make things a little bit easier for you. But if you're starting from scratch like we are today, we are building a cohort from scratch, we can simply clear this filter so that we get the entire set of data to start working from. So I'm just going to click clear right now. Okay, so it's cleared. And you can see I now have the full breadth of the GDC data available. We do have 84,000 cases across the entire portal. But let's actually start narrowing down to what we want today. So we know we are looking for bronchus and lung cancer patients. So in my Cases tab under Primary Site, I'm going to select bronchus and lung. And you can see immediately the results have narrowed down. And you can tell this not only by the changing of the table, but if you look at the counts in the tabs, you'll see the numbers change. So it went from 84,000 down to 11,916. Now, pro tip, if you look at the options on the left-hand side, the number that you expect is actually already displayed. So you can see here, bronchus and lung, it has 11,916. It's a little small here, but this little gray box. And I know that if I instead picked breast, for example, there's 8,897. So let's try that right now. I'm going to uncheck bronchus and lung. Goes back to 84,000 total. Now, if I click breast, I should expect that number to change to 8,897. And it does. Okay, so it's just a little visual indicator you can use to, just to make sure things are going as expected. So let's go back now and select bronchus and lung. Okay, 11,000 cases. I want to narrow this down further because we're interested in adenocarcinomas. So I'm going to scroll down to the disease type property, and I'll pick adenomas and adenocarcinomas. So this narrows down now to 8,343. Now, there are more conditions I wanted to add. Let's use clinical information. We wanted to look for females who are affected by this type of cancer. So I'm going to select female on gender. Now it's narrowed down to 4,783. Okay, I continue to refine my cohort to what I want. And then we also wanted to specifically look at females diagnosed from 45 years of age and up. So I will go down to the diagnosis set of properties. I will select age at diagnosis. You can see here I can actually specify that age in years or days. We'll keep it simple. We'll keep it in years. So from 45 years to, now I'm not going to set any maximum. If we leave this blank by default, it'll be basically 45 and up to whatever maximum is available in the portal. Click go here. Okay, I have refined my cohort. This is exactly what I want, and I can see there are 1,474 such cases in the GDC. The nice thing you'll see is as I was clicking through those filters, it was actually building my query or building my, building my search. So if you can read up top, it says, gender is female, age at diagnosis greater than or equal to, this is displayed in days, but if we converted that back, it'd be 45 years. Disease type is adenocarcinomas, and then the primary site is bronchus and lung. So we're just going to take a quick swig of water. Sorry about that. So you can see as you build it, it's nice because you can validate that your query is being built correctly. So now that I have the results, I can just do a quick scroll through just to see if I'm still interested in this data. So right here, we show 20 results at a time on the page. If you want, you can actually extend that up to 100 so you can see a little bit more. 100 is the maximum per page. So that's just a little extra convenience afforded to you. There are other things that you can play with just to make this view a little bit simpler. So I can arrange my columns. So for example, age at diagnosis was something we were very interested in. By default, it's not in that default view, but maybe I want to add it to the view just to make sure things are what I want. So I'm going to click AGIC diagnosis here. And you can see on the right-hand side, it actually shows me the specific age of each of these patients. Additionally, I can also do some sorting on the table. Now, the sort options are a little bit limited right now. We may be looking at expanding that in the future, but it's just another convenience that is provided to you. Now, the rest of the buttons in this toolbar are related to data download, so I will cover that a little bit later. Additionally, there's a few visualizations on the other tabs if that will help in your decision-making process. So maybe I want to quickly go to the Genes tab. And you can see there's, for these 1,474 cases, there are 20,675 genes that affect them. And you can see there's a nice little bar graph that shows you the genes that are most frequently mutated across this cohort. So again, as you build this cohort, all of the visualizations here will be contextual to that cohort. So you can see here, if I scroll down and I look at TP53, it will say in this first column here, TP53 in this cohort, there are 128 cases affected by TP53. So I won't go through all of the columns today. We have good documentation on that. But you can see you can use the tools as you build the cohort to continually validate that, hey, this is what I want to look at. Okay. I'm going to go back to the Cases tab. So now that I've finally built my cohort, I actually want to save it so that I can do some analysis on it. So how do we do that? Well, there's a button to the rightmost of the toolbar here. It says Save or Edit Case Set. Again, a set is a group of things that we want to save in the portal. Now, I could be very, very prescriptive. I could say, well, you know, I actually only want to see the top 5 cases because they have the most mutations. And I could specifically select these 5 rows and then save these 5. But today we're going to look at the entire cohort. So I don't need to select anything specific. By default, if I just click the Save Case button, it will allow me to save everything that I've built. So I'm going to do that right now. I'm going to save as new case set. So by default, it lists the filter properties that you've added. So it's a nice convenience. I will make it a little bit more descriptive just because while this is useful, it's not the most human legible. So I'll just say bronchus and lung adenocarcinomas. And I will say females diagnosed greater than or equal to 45 years. Okay, and I'm going to save that. And you see there's a little message that says, hey, you have saved your sets. And then you're probably wondering, well, now that I've saved it, how do I use it? Where can I find that set? Let me just reduce the screen space here so you can see a little bit more. So there's an area called Manage Sets, if you look at the top right of the toolbar. And this is where all of the saved sets, whether they're cases, genes, or mutations, are kept. So if I go here, you can see I've actually created a couple in advance, but here's the one that we just created, bronchus and lung adenocarcinomas for females diagnosed greater than or equal to 45 years. So any cohort that you would save will be saved here. You can see that the type of information saved is cases, which is good because we didn't save any genes or mutations. Shows you the number of items in your cohort. And there's you can also export that list of cases as a TSV. We'll get to that a little bit later. Okay. So that sort of that finishes the first step in the process, which is explore the GDC, build a cohort. Now that we have a cohort, what type of analyses can we actually do? So for that purpose, I'm actually going to create another cohort first so we can do a little bit of comparison. So I'm going to create the same cohort except for males diagnosed 45 years and up. So this is a little bit of deja vu. We're going to go back into the portal. I'm going to clear the cancer gene census filter because I want to look at everything. Again, I will pick bronchus and lung. I'll pick adenocarcinomas. This time I will select male instead, so you can see the numbers are different. And again, I will enter 45 years and up for age of diagnosis. Okay, so there's 1,270 such cases, and I want to save them all. So I'm going to save this one. Let's name it similarly. So bronchus and lung adenocarcinomas, males diagnosed greater than or equal to 45 years. Save that. I will go back to my Manage Sets area. Great, now I can see both of these new cohorts are available. So the GDC has some very interesting out-of-the-box tools for analysis. The main ones are in this analysis area up at the top here. So I'm going to click analysis. And we have 3 right now. We're actually planning on rolling out a few additional ones later in the year. So stay tuned for that, a little bit of a marketing teaser. But for now today, we're going to go through these basic ones we have here. So the first type of analysis we have is called set operations. And basically, this allows you to basically compare the numbers between 2 cohorts and see if there's any overlap. So as I mentioned earlier, we want to compare the female cohort that we created and the male cohort. So if I enter my set operations, I can compare up to 3 sets of the same type. Today, of course, we're looking at cases, so we're only going to focus on that. And we're only going to do the simple example of comparing 2. So I'm going to compare my female one and my male one. So I've selected them, and now I will click Run. Ta-da! So you see there's a very simple Venn diagram that got displayed. What does this Venn diagram actually tell me? So it's basically saying set 1, S1, are my females diagnosed 45 years and up, and set 2 are my males diagnosed 45 years and up. And in the middle of the diagram is any intersection between these. Now, I chose this example very specifically because we know what the result would be. The result would be 0 because there would be no overlap between those 2. So this is correct. It's showing me 0. So great. I know that they don't overlap, and maybe there isn't anything more interesting for me to explore. So you are probably wondering, well, that doesn't seem very useful. Can you show me a different example where this type of analysis might actually make some sense? Well, sure. If I go back to the sets that I created before we started the webinar, I actually created 2. The first one was bronchus and lung adenocarcinomas for drinkers, so people who have reported as having been frequent or occasional drinkers. And then the second one, again, bronchus and lung patients who are smokers, who have reported smoking, either current smokers or reformed smokers. Now, why would this be interesting? Well, perhaps I am looking at exposures, so different things, lifestyle exposures, drinking, alcohol, weight, BMI, that different cohorts have been affected by, and I want to see if there's any overlap between drinkers and smokers affected by this type of cancer. So I'm going to go back to analysis. This time I'll select the first 2. So there's 99 drinkers, 62 smokers. I will run. Oh, so it's interesting. This time the results are different. You can see there's actually an intersection of 53 cases. So there's 53 bronchus and lung adenocarcinoma patients who are both drinkers and both smokers. So perhaps I have a specific scientific or research question where I actually wanted to look at the intersection of these 2 groups as well as those 2 groups individually. So this analysis helps me narrow down those numbers. And if I wanted to, I can actually save that set. So you can see when I select that intersection, there's a little button on the right. It actually allows me to save that intersection as a new set. So maybe I can do more analysis on this refined cohort. So that's sort of the power of the set operations tool. So now we'll move on to the next tool, which is the cohort comparison tool. So this tool allows you to take two cohorts and do a very basic comparison of some of their high-level properties. Now, I wouldn't say it's a very detailed or advanced comparison. That's something that you can definitely be encouraged to do offline with maybe your own tools. But we do provide a sort of a high-level side-by-side look. So again, what you would do is you have to first select the cohorts that you want to look at. We'll go back to our example. Let's compare the females and the males that we created. We'll do a run. Okay, and it generates a set of visualizations that compares the data. So you see right off the bat, we have a survival analysis comparison. And it's quite interesting. It just so happens for these two populations, the survival analysis, if you look at the curve, it's actually quite similar. Now, I'm not a scientist. I can't say if there's anything particularly important about that, but these are the kinds of things that the tools help you look for. So you might say, well, the survival analysis is quite similar for these. I want to investigate more of their clinical properties, for example. On the right-hand side, you can see as a convenience, we actually show the set operations comparison that we had earlier. So if you wanted a quick glance, you could actually use the cohort comparison as well. So again, you can see there's zero intersection between these cohorts. And then we provide some very high-level clinical properties that you can compare. Again, we do look in the future to add some more properties that you can select here by default. But by default, we show you 5 high-level ones. So you can see the gender bar graph. This one is fairly obvious. We know that the first cohort is 100% female, and the second cohort is 100% male. So that's not too interesting. But if I go down to the vital status, so you'll see this is sort of data completeness. How many of these patients had their vital status reported. You can see quite a few were actually not reported or unknown, but there are some alive and some dead. And then we look at age of diagnosis. Now, this is quite interesting. If you look at so the ages are broken up in chunks of 10 years, so in decades, from 40 to 49, because that's where our starting age fell, and then all the way to the maximum. But you can see that the numbers, the percentages, are actually quite similar. There are small differences, but if you look at sort of the comparison of the bars, again, fairly similar. So I can't, again, say if that has any scientific significance, but these are the trends that you can use the tools to look at to lead you in, future directions of, exploration. So now we'll go to our final analysis tool. That would be the clinical data analysis. I won't go through this one in too much detail. We did have an excellent webinar in February where this was covered. So again, I encourage you to check out our archives, but I'll do a quick refresher here. So this tool lets you look at specific clinical properties for that cohort. Now, this isn't a comparison, so I can only select one. So we will look at our female bronchus and lung population, and I will run. Okay, and it has loaded. So quickly, you'll see that the structure of this is a little similar to exploration. It's not quite filtering your cohort, but there is a control panel on the left-hand side that lets you enable different clinical properties. You can see that they're grouped in the same order as in exploration. So demographic properties first, diagnosis properties next, treatment, exposure. And by default, this is very similar to the cohort comparison you saw. So consistency is key. By default, we show ethnicity, gender, race, age diagnosis, and then we also show the primary diagnosis. The nice thing is that all the visualizations are color-coded, so you know what type of clinical property you're looking at. So for example, the demographic properties are all blue. So you see these bar graphs are blue, and then the diagnosis ones are orange, so these ones are orange. If I wanted to look at treatment, so maybe I want to look at the treatment type that these patients have undergone. So I click treatment type. You can see this one is color-coded green. So each of these properties will open up a new card. If I want to look at exposures, maybe I'm interested in their weight. So if I enter the weight, this is actually in kilograms. We do have a data dictionary that does explain these units, and we do look at some improvements such as exposing those units as well on the interface. But again, you can see weight is also color-coded. So it's just a really quick and easy way to dissect some of the clinical properties of your cohort. You can see the first card is also a repeat of the survival data. So although this cohort has 1,474 cases, only 272 had survival information, but that is available here. And each of these cards can be downloaded in graphical format, SVG or PNG. So you could, for example, include in a publication or a paper. So that's clinical analysis. The final type of analysis that I will cover today is actually in the exploration page itself. So I'm going to go back to exploration, and you kind of saw this a little bit earlier. The reason some of the visualizations are in exploration itself is that you can actually reference that information as you build your cohort. So it just gives you a little bit more information to help you narrow down. So on the cases tab, there's some pie charts that gives you a little bit of a breakdown. So right now, so I'm just going to clear this so that we see all of the data across GDC. Okay. We can use our cohort a little bit later, but I just want to show a larger wealth of data. So I'm looking at all 84,000 cases. I can see a breakdown by primary site. So 11,000 bronchus and lung cancers, 8,900 hematopoietic and reticular. I'll save the pronunciation for another time, but you can see there's a breakdown of all of these properties. Earlier I had mentioned the genes, so you can see the most frequently mutated genes for that cohort. Again, we repeat the survival plot. So again, a lot of high-level information to help you make your decisions. And then the most significant analysis on exploration itself would be the OncoGrid. So for the OncoGrid, I'm actually going to use our cohort just because to load the OncoGrid for the entire GDC will take a few seconds. So let's just go back to the set that we had before. You can see there's a link on the right. I'm going to view the female cohort in exploration. Okay, so it's loaded up the 1,474 cases we saved. And now I'll load up the OncoGrid. Okay, so here we have a pretty standard OncoGrid. We do expose the top 200 most mutated cases against the top 50 most mutated, the top 50 genes by mutation. Just going to go into full screen mode so we can see it a little bit better. Again, we won't cover OncoGrid in great detail today, but you can see I can filter the grid by specific mutation types. So earlier I had mentioned maybe I'm only interested in missense mutation. So I'm going to uncheck all of these. And then it reloads, and I've narrowed it down just to missense consequences. Maybe I'm not interested in copy number variations today, so I'm just going to remove that. I only want to look at the somatic mutations. And here we are. You can see at the bottom there are some very standard data tracks that help me identify some properties of the case. So each of these columns is really an individual case. It's a bit small, but you can see here, I can see the race of each case, the general range of the age of diagnosis, their vital status, so on and so forth. Again, this OncoGrid can be downloaded in graphical format to include in your publications and papers as well. Okay. So that completes all of the basic analysis types that we have available. So we've gone through building your cohort, analyzing it with different tools. Now we'll get to downloading data. I'm just doing a quick time check. We have about 20 minutes left. Okay, that's great. Should leave us some room for questions. So data, so there's, I would say, two main types of data that you can download. One would be sort of the raw source data, whether that is clinical information, it could be the raw genomic data. And then there's also visualizations. You want to download the charts and the graphs for your papers and publications. So we'll go through each of those in turn. So I'm going to go back to the Cases tab. Again, we're looking at the cohort that we've built. Going back to the toolbar that I had mentioned earlier, there are 4 key buttons here. The first is the biospecimen button. So this allows you to download all the biospecimen information specific to your cohort. So that would include things like the samples, the aliquots, the analytes. I won't download all of these right now, just because that it could be time-consuming just based on the size of the file. I have downloaded some of these in advance, so I can give you a quick snapshot. The clinical button is to download the clinical information that we looked at earlier, so the demographic information, exposures, treatments, so on and so forth. The next 2 buttons are not for anything specific. The JSON button allows you to download this table that you see here in JSON format for all cases in the cohort. The TSV view, however, is only to export the current view in TSV format. So right now I'm showing only 20 cases. So at most, as I mentioned earlier, I could export 100 of these cases in TSV. Currently, we don't allow exporting the entire cohort as TSV, but if you do require that, you can do it through JSON. So I won't look at these 2 files because you do see the data already on screen. It's really a replication of what you see here. But I will give you a quick example of biospecimen and clinical. So I'm just going to go to my file browser. I do use a Mac, so apologies to Windows or Linux users today. If I look here, the biospecimen, let me expand this a little bit. So the biospecimen gets downloaded as a tar because there's multiple files. You can see in this folder, it gives me all of the aliquot information, analyte information, portion information, sample, and even the slides that are associated with the cases in this population. So I'll just do a quick example. I'll bring up the sample TSV and I'll open it in Excel. Okay, so you can see there's a nice spreadsheet that you can use. It has all of the information. There's a lot of columns. Again, we have documentation on the meaning of these fields. So I can definitely refer you to those links after the presentation. So that's an example of biospecimen information. And then if I look at clinical information, again, there's some basic demographic information. There's exposure information, family history. If there is treatment data, that would be available as well. Let's take a look at exposures as an example. Again, very similar. The listing of all the cases, And some of the properties I mentioned earlier, you know, their alcohol consumption, their smoking history and patterns, so on and so forth. Okay. I'm going to go back to the portal now. Now, the other thing that I mentioned was downloading of visualizations. Now, each visualization in the portal has its own download button because it's specific to that chart. There isn't a button that allows you to download everything at once. You do have to be prescriptive in what you want. So, for example, if I went to the Genes tab and I wanted this most frequently mutated genes bar graph in my paper, you can see there's a little download button at the top. I can download the SVG or the PNG, or if I want the raw data to maybe generate my own visualization with some other tool, I could download the JSON format. I'll give you just a quick example of what that looks like. So I've downloaded that in advance. So here you can see the PNG is exactly as it was on the portal. So you can do this for each chart that's available. Just as a quick example, I'll go to OncoGrid as well. I won't print it out, but you can see there's also a download button, again with the same formats. If I go back to the clinical analysis, because that's also very interesting. So I'm going to run actually I think I have I have my analysis saved from beforehand. Again, each of these cards has a download. But as a convenience, because the clinical analysis we find is actually quite popular and quite useful, we actually have a button that allows you to download all the images as well in either format. Again, I won't do that today. Now, the final type of data download that we haven't covered is downloading the raw genomic files associated to your cohort. So we'll quickly do that right now. Quick time check, about 15 minutes. Okay, should be okay. So I'm going back to, apologies. Let me bring up our cohort again. So here's our female bronchus and lung cohort. You'll notice that there's a button at the top of the Cases tab here. It allows you to view all the raw genomic files associated to this cohort in our file browser, or what we call the repository. So I'm going to click that now. And you can see up top, it has taken me to a different area of the portal. Now, the case, we know this is correct because the cases, it's the 1,474 cases we were looking at before. And now we can see there's 12,208 files associated to those cases. And the repository is structured the same way as exploration. I can use the filters on the left-hand side to narrow down the files I want to look at and download. Downloading the files is a little bit of a different experience. It's more like an e-commerce experience. You would add these files to a shopping cart, and when you're ready, you basically check out and download those files to your local computer. So we'll use a very simple example right now. Let's say I just want to look at some specific math files for these cases. So you can see data format is one of the filter properties that you can use. So I'm going to select math. You can see there's 247 files. And let's say I am interested in just the controlled and open math files for two variant callers. Let's say Muse and Mutex. So I'm going to take the open file from Muse, the control file from Muse. So you can see there's a little shopping cart icon, just like any good e-commerce site. I'll take the open Mutex file and then the controlled Mutex file. Now, controlled files are basically files that are access controlled. You must have db/gap access and authorization for that project to have access to those files. So if you haven't applied for access, then you would have to do so before you can actually download them. But let's assume today I already have access. You can see my shopping cart after I've clicked this button. In the top right corner, it actually shows my cart, and it has 4 files added. So I'm going to go to the cart right now. Okay, and the cart's really nice. It gives you a nice breakdown visually. You can see the number of files I have on the left-hand side. the total download size, in case that's relevant to you. And you can also see on the right here, there's a table that shows the number of authorized and unauthorized files. So 2 and 2. As a convenience, you'll see some additional download buttons here as well. So for these specific files, I can also download the specific biospecimen information or the clinical information, similar to the buttons you saw earlier in exploration. Additionally, I can also download a spreadsheet of just the samples, or I can also download the metadata associated with these files. Again, I won't go through these examples in too much detail. Feel free to explore these on your own time. But let's actually download what I want from this cart. Now, you'll notice right now that I actually haven't logged in at all into the site, so it actually doesn't know who I am or what I have access to. So what happens when I try to check out? So I click this download button, and I will download cart. Now, one quick point, you can actually download a manifest. For example, sometimes the number of files or the size of files may be too large to download through the web. You can download a manifest that you can use to download those files through the data transfer client tool. So we actually had a webinar last month on the DTT tool, so this kind of ties into that one. So that's what the manifest is for. But today we are downloading through the web. It's only 1.19 gigabytes, so that should be all right. So when I click download, you can see I'm actually not allowed to download all of them. It says I have 2 files that I'm authorized to get and 2 files that I'm not authorized. And you can see the button says I can only download 2 of them. Now, if I want to download everything, I need to log in first. So there's a prompt here to log in. I will log in now, apply my details. Once I've logged in and I try my download again, you'll see that the pop-up has changed. It's now saying I can download all 4 files, but I have to first consent to the GDC's data use agreement. So we'll assume that I am a law-abiding citizen. I will consent to this. The download button is now enabled, and I can click download, and these raw files will be sent down to my laptop, and I can continue my analysis further on. Okay, so that leaves us with, apologies, went over a little bit. There's about 8 minutes left. So that brings to a close the webinar today. Just to summarize, we went through building your custom cohort, analyzing it with our out-of-the-box tool, and then downloading raw data for further investigation offline. So I'm going to close this off and hand it over now to any questions.
Thanks, Brandon. I know you're having a little trouble hearing us in particular, so we'll have to go through these questions.
Let's stop screen share here.
So the first question is, Genie program data is not available for exploration, correct? And the answer is correct. Right now, the Genie data is controlled access. We are working on a feature in the GDC to allow a user to select a controlled access.
See, there's quite a few questions in the chat. So how do we download the 20,675 gene results from that page? Let me share my screen again. Apologies for that. So let's go back to our cohort. So the genes tab, if you're interested in these 20,000 genes, again, we do provide 2 buttons here. One is the JSON download. The JSON download will allow you to download all 20,000 of those genes. And we do, again, have the TSV export, but that is only for the current view up to 100. So I would advise, if you wanted to analyze this entire set of genes, you will need to download the entire JSON. I can download that right now. It may not take too long. If it finishes before the end, I'll show you what that looks like. Go back to the analysis. Go back to my original analysis, which had all of the data. So this is my 1,474 female lung cancer cases. we are interested in only a specific set of specific values of certain clinical properties. So for example, I only want to look at 3 specific races. So let's say white, Black, African American, and Asian. The clinical analysis tool actually allows me to save this narrower set of cases for a new analysis. Unfortunately, I can't filter it right in the analysis itself, but I can save these 241 individuals as a new case set. I'm going to save this as, let's just call it, smaller case set, just in the interest of time, because we're coming up on the end. I've saved that set. If I go to Manage Sets, I can see that it's there, 241. I can then go back to my clinical analysis and select that smaller set to visualize. So I'll run it again. And when I scroll down, I can see that the race properties has now narrowed down to just those 3 values. And I can continue my analysis just on this reduced population. But that is a very good enhancement that we may look at in the future, maybe allowing some more filtering on the cards as you go. One more question. Is it possible to isolate cases by mRNA expression of a specific protein? So that should be possible. So I'm just going to go back to exploration here. Now, what you may have to do, because I think microRNA expression, let's see, it should be one of the genomic filters. So if I go to genes, there isn't, let's see, biotype. So there's microRNA. So I could do, so I may first look at a specific gene, so TP53. Again, this may not be a scientifically relevant example, but I can narrow the population down to that gene. You see the biotype, it did not have microRNA expression available. What you may have to do, and it's a little bit roundabout, but you can actually take this set of 713 cases that are affected by TP53. I can view them in the file repository, and the repository will let me see which of these cases have gene expression files associated with them. So you can see under Files, so I've actually basically transferred those 713 TP53 cases to the repository. Under data type, you can see that there are actually gene expression quantification files associated. So I can actually narrow that down, and you can see the cases here has gone from 713 to 638. So unfortunately, it is a little bit more roundabout to narrow it that way, but it is possible. And then what I can do is I can then save this set of 638 as a new set. So maybe TP53 with gene expression data, and I can save that, and I can use that that new cohort for further analysis. So excellent question, and actually gives us an idea for some future enhancements as to maybe make that process a little bit smoother. Next question, can one retrieve expression data from the GDC data portal for integrated analysis across different studies? Yes, so if we go back to that, so I will assume that we are talking about downloading raw file data. So in our file browser, so let's stick with this example. I'm just going to clear the set that we brought in, which was specific to TP53. Let's just look at gene expression quantification files. So there's 14,000 of those. But I want to look at the raw data across different programs, let's say, or different studies. Again, if we use the filters on the left-hand side, so this is the file filters, but if I look at the cases tab, you can see a repeat of the properties that you saw in exploration. So I can actually narrow this down by the program. So maybe I want to look at only TCGA and target gene expression files for breast cancer. Oh, sorry, target doesn't have any. So let's say TCGA and CPTAC breast cancer gene expression files. And you can see there are 1,226 cases that this applies to, and a total of 4,201 files. Now, this is a lot of files. The file sizes are a bit small, but I don't quite recall, but there may be a limit on the cart as to the number of files. So this is a nice dovetail to the DTT webinar. I may need to add all of these files to the cart, and I may need to download my manifest, so I can use that manifest to download in bulk through the command line transfer tool. So you can see the manifest got downloaded. Here's the manifest, and maybe I can supply that through the tool. Thanks, Sharon.
Okay, I'll take the next two questions are related. They're about dbGaP access. One question: I was approved from dbGaP access for the control math. If I was approved, can I let my team member analyze the math file? And the other one's also related: if I apply for access to data as a PI, can my students do the download, or do I download and then share? The way dbGaP works, and I actually apply for dbGaP access as well, is I specify my team members in collaborators area in dbGaP. And so once I'm approved, anybody I have listed as a collaborator from my team is able to access the data. So you have to make them collaborators to the team, to your team, and then you also have to give them download access through dbGaP as well. And there's a separate button that allows you to do that. But they need to be the collaborators. Okay, and let me go ahead and if everybody could please post all your questions in the Q&A and not the chat. I'm actually using the chat to communicate with Brandon right now, so I'm definitely missing all your questions in the chat, so please post them in the QA. And these are fantastic questions, so I really thank you.
Okay, so does GDC have all the mutation data available from all the cancer databases available? So what we have available is from the programs that have applied and submitted to us. Again, you can see that program list in the exploration. I can't make a blanket statement and say we have all the data from all the cancer databases, but we do have a wide wealth of studies available. So if I look at, this is actually nice because I can show you a different area of the portal. If we look at the projects area, we can see a breakdown of what programs have submitted to us. So you can see that on the left-hand side, there are quite a few programs, some big names, TCGA, FM, Genie. We are also continuously having new programs on board and new data being submitted to us. So I would say, keep an eye out, keep up to date with the portal and our releases, our data releases. We're always getting new data, and we're trying to accumulate and curate, all the best data. Can you analyze a specific set of genes? Yes, you can. Again, going back to our exploration tool, it is quite powerful. So if I go to the Genes tab, I had mentioned that you could search by a specific gene, but actually you can supply a list of genes. So you can see here there's a button that says Upload Gene Set. So I may be interested in multiple genes that impact breast cancer. Now, what I can do when I click this, it gives me a little pop-up. I can either manually type and paste the list of genes, or I can upload a file that I've pre-created with that list. But let's try that right now. Let's say, TP53, CRAS, and BRAF. So it validates that these genes are available in the GDC. That's great. It's match 3, and I can submit this. And then again, my search results get narrowed down. You can see just a quick eyeball, my three genes were accepted, and there are 17,595 cases that apply. And then if I want, I can save these cases, right, for further analysis, or I can save the specific genes for further analysis, or I can save the specific mutations. So again, it all ties back to the concepts we were talking about today about saving sets and then supplying those sets to different tools throughout the portal. Yes, please do reach out to our user support. We are usually quite responsive, but we're happy to answer any questions you have to help you further your use of our portal. Is there a way to link genomic data available in GDC with the SEER database? By link, do you mean linking the visualizations and the query results as you explore? I wonder if we can get a small clarification on that, because that would not be possible today. Maybe something that we would have to look at in the future. We don't have any integration with the SEER database today, I don't believe. Sharon, please correct me if I'm wrong.
Yeah, we don't have integration with the registry database yet. It's something certainly would be a good future feature to look into. There was a question on authorization needed to publish research made from open access data. We don't have any specific authorization required for open access data. We just, also certainly ask if, based on the data sets, it's that you follow the data use agreement that you signed as part of the DPGAP registrations.
So how to restrict driver genes? You would need to identify those driver genes as a gene set or list that you're interested in. We do not have a specific filter or property today that says, for this type of cancer, only filter based on these driver genes. You would have to supply that gene set or list yourself through the method that I showed you just now. But again, an excellent idea for some future enhancements. Maybe we could have a new filter or a new mechanism to say, for specific cancer types, only narrow down to X, Y, and Z predefined driver genes. But right now, those would have to be manually applied. You generate customized cohorts within the same project based on the expression level of a gene set. And so that is not available currently. Later in the year, we do have a new gene expression analysis type coming out, where you can visualize expression values for a cohort. But currently, that is not available today. But it is a feature that is being rolled out later in the year. When we investigate a gene set, the sample number is different from the whole gene set. How to understand number difference. This one I am not as clear on the answer on. I'm wondering, maybe Hamansu, if you're online, would you be able to maybe field this one, or perhaps Bill?
Hi, Brendan. I think it might be because of the consensus gene. I don't understand the question either, but.
The next question is on survival analysis.
What is the survival analysis? Yes, so the cases, yes, the survival analysis is for the 17,595 cases affected by these three genes, of which there are only 5,721 that have actual survival data. So again, the visualizations are always contextual to the cohort that you have built. So what that means is the number of cases that you see in this total here, that is what the charts and graphs will be.
Also, if those cases don't have any survival data, so they won't get charted.
Do you have any plan to deposit the ICGC data? So that is a data submission question. I will hand that one over to Bill if he's still online?
Yeah, Bill had to run to another meeting. All right, I think I'm just going through the list of questions, and I think we covered all of them on the Q&A. Just looking at, sorry if folks put them on the chat because I'm actively using the chat right now, so I do apologize if I missed any of your questions. And I do thank you all for staying on, this webinar so late. I think this has been a great exchange and gave us some wonderful ideas for new features. So we'll end this webinar session. Thank you so much, Brandon, for the wonderful demonstration and presentation. And you all have a great rest of the day and stay healthy. Thanks, everybody.